Very good morning to all of you on this Monday morning. Today, we are going to start again a new topic again a new pathway that is associated with metabolism. Last lecture, we have seen glycolysis, which was an excellent part that how reduction of sugar would take place and it gets converted into pyruvate. A similar pathway is there by which that particular sugar can be processed into a different product. So let us understand this topic of ED pathway. Now, this ED pathway of glycolysis, this ED pathway is very similar to glycolysis. So, glycolytic pathway is the most common route for the conversion of hexose to pyruvate. We have seen last lecture. Another pathway which is similar to has been discovered. 6-phosphogluconate is 6-phosphogluconate is dehydrated to form 2-keto 3-deoxy 6-phosphogluconate or KDPG. It is also called as KDPG. The key intermediate in this pathway. KDPG is then cleaved by KDPG aldolase to pyruvate and glyceraldehyde 3-phosphate. The glyceraldehyde 3-phosphate is converted to pyruvate in bottom of the portion of the glycolytic pathway. So it will run only the glycolytic pathway, but when it comes in terms of pyruvate, at that time, there will be separation of this pathway that would take place. If the eternal Dwarov pathway, that is ED pathway, degrades glucose to pyruvate in this way, it yields 1 ATP, 1 NADPH or 1 NAD per glucose metabolism. So ultimately, it will generate ATP molecule by reduction of that particular sugar after the pyruvate formation. So most bacteria have the glycolytic and pentose phosphate pathways, but some substitute the ED pathway for glycolysis. The ED pathway is generally found in organisms such as Pseudomonas, Rhizobium, Azotobacter, Agrobacterium, and few other gram-negative genera. Very few gram-positive bacteria have this pathway, with Enterococcus faecalis being a rare exception. So basically, this pathway is generally being followed by selective organisms only, but organisms which belong to gram-negative organisms are mentioned over here. Some exceptions in gram-positive are all there. For example, Enterococcus faecalis, which is the only organism wherein this particular pathway has been formed or has been observed. While on the other hand, the glycolytic pathway and the pentose phosphate pathway is generally been utilized. See here, glucose to glucose 6-phosphate to phosphogluconate. Now, from this phosphogluconate, 2-keto 3-deoxy 6-phosphogluconate is born. This is called as KDPG. This KDPG will lead to pyruvate as well. Once it goes to pyruvate, it can go to the tricarboxylic acid cycle as well. But when it comes in terms of glyceraldehyde 3-phosphate, once it comes to glyceraldehyde 3-phosphate, this glyceraldehyde 3-phosphate, it gets converted to 1,3-bisphosphoglycerate, 3-phosphoglycerate, 2-phosphoglycerate, phosphoenol pyruvate, and pyruvate. So ultimately, from glyceraldehyde 3-phosphate, which is a product of this pathway, this glyceraldehyde phosphate gets converted back to pyruvate only. But during working in this particular flow, it will generate 2 molecules of ATP. This is the important part. Degradation of that sugar would take place and generation of ATP would take place as well. Now, from this mechanism also, glucose will be degraded to glucose 6-phosphate, similarly here. Now, this pyruvic acid or the pyruvate which is generated here can go with acetaldehyde plus CO2 and ethanol is being produced. So this pathway is generally been utilized during fermentation procedures and formation of ethanol would take place. Now, these are the organisms which are there, which can follow both the pathways. For example, Agrobacterium species are there, which can Arthrobacter, which can follow EMP as well as EDP pathway for the process that would take place. There are a number of organisms also that come in play or that are helpful. Basically, strictly ED pathway producing organisms are working on these organisms are Pseudomonas species, Rhizobiums, Thiobacillus, and many more species are there, wherein they work out with ED pathway only. Key enzymes. What are the key enzymes of the ED pathway? Glucose 6-phosphate gets converted to 6-phosphogluconate. to 2-keto-3-oxy, and all these enzymes are there. For example, glucose 6-phosphate dehydrogenase, 2,6-phosphogluconate dehydratase, 3,2-keto-3-deoxy 6-phosphogluconate aldolase, as in these are AMP, ultimately in the EMP pathway. So NADP dependent glucose 6-phosphate dehydrogenase converts glucose 6-phosphate to 6-phosphogluconolactone converted to 6-phosphogluconate. 6-phosphogluconate dehydratase removes water molecule. See here, water molecule has been removed from 6-phosphogluconate and 2-keto-3-deoxyphosphogluconate, that is KDPG, is being generated. KDPG aldolase splits KDPG into pyruvate and glyceraldehyde 3. This is the reaction. Glyceraldehyde 3 phosphate and pyruvate is being generated. And the key enzyme of AD pathway are 6-phosphogluconate dehydrogenase and KDPG aldolase. So this ultimately, this key enzyme, it gets converted into pyruvate then again. Now, glycosylate cycle. This is again an important point that is to be considered. Anaplerotic reactions. To meet the demand of carbon requirement, the cycle operates from gluconeogenesis. Modified TCA cycle found in plants and some microbes utilizes fatty acid as a source of energy in the form of acetyl coenzyme A. In this cycle, CO2 evolving steps of TCA cycle were bypassed, and instead acetyl coenzyme A is being utilized, which condenses glycosylate to form malate. Succinate is a byproduct used by biosynthesis, particularly gluconeogenesis. So this is an alternate pathway that comes in play when it comes in terms of citric acid. My next presentation will be based on the TCA cycle itself, where we can understand this particular point. Correct? So today we have learned in terms of ED pathway. This is the glycosylate bypass or glycosylate cycle. This TCA cycle we are going to learn in our next lecture. TCA cycle consists of acetyl coenzyme A to citrate to isocitrate to alpha ketobutyrate, succinyl coenzyme A, succinate, fumarate, malate, and oxaloacetate then again. So this completion of the cycle would take place. This glycosylate bypass goes with isocitrate and directly gets converted into malate. Forms within one molecule of acetyl coenzyme A, it gets converted to malate. So this is in bypass cycle that comes in play, called as glycosylate cycle. See here, certain key enzymes. So today we have learned another pathway called as ED pathway. In my next lecture, we will continue with some other pathway of this particular topic. Okay. Sure. Thank you.